So you have very steep mountains, steep canyons. So for GEDI as a large footprint instrument, that's a challenge, but we will see how well it will go. So what I would like to do is to be able to capture this type of diversity and map it so we can monitor it over time and we can also protect it. This is showing the whole study area. It's over 7,000 square kilometers covered by the Airborne Snow Observatory. So we have airborne LiDAR data, wall-to-wall coverage. The colors here just kind of show different structural traits. So you see the different structure types. For example, different types of vegetations here on these slopes in the valleys versus these plateaus. You see these big canyons cutting in here. So we have, for context, Yosemite National Park is up here. Some people might know it. This is mainly Kings Canyon. Sequoia National Park is also partially covered. So there is some pretty tall sequoias over here. So if you see a canopy height of 80 or 90 meters, that's not an error. That's actually in the study site. So can we measure this from space? Can we measure structural traits and structural diversity? We tested this using the GEDI simulator. So Steve Hancock has developed this method to use an airborne point cloud and to simulate how GEDI data will look like. So we used that to find out the potential of GEDI to map those kind of patterns. So that's how it looks like if we look at the simulated pattern. Basically, this is like different overpasses from the ISS. So you get this kind of transects. So this is the type of underlying airborne point cloud. It's pretty dense, mostly more than 10 points per square meter over the whole area. You see how those GEDI footprints kind of lie within that forest. And this is how the coverage looks like with data simulated for 2 years. So that's how much data we expect after a 2-year operation time. And it's the full coverage, so here we don't simulate cloud cover. So in the results you will see the difference between just a theoretical simulation of all the overpasses and what happens if we actually try to simulate cloud cover of the area. So how do we measure functional diversity or structural diversity? In this case we look at a range of structure metrics, or I call them traits. Those are the well-known ones, the relative height values of 25, 50, 75, 95 percentile and maximum canopy height. Then we look at plant area index as a density measure. We get that for the different layers every 10 meter, but also for the total column, so total plant area index. Then we have what I call canopy ratio. So that's basically just in this case, I use a ratio between the 25th, the distance of the 25th to the 95th percentile, so basically the crown depth, to the total canopy height. So if you imagine a sequoia tree, that would be the extreme case. You have a very tall tree but a short canopy, so then canopy ratio would be very low, whereas a very densely packed canopy would have 100%, for example, canopy ratio. And then we have foliage height diversity, which tells us about how many vertical layers do we have and how different are those layers to each other. So it's like a Shannon diversity calculated on the layering of the canopy. Now from there we get like a whole range of traits. These are now the traits compared between the airborne laser scanning derived traits and the GEDI traits. So up here are all the height-related metrics. Then we have the plant area index for the different layers, plant area index of the total column, and so on. So on average, GEDI is doing pretty well. Most of the traits are well predicted or measured. This is the average value for slopes lower than 25 degrees. And we see that if we just look at height, density, and layering, we get R-squares of around 0.6, 0.7, and for flatter areas, it actually goes much higher, like 0.8, 0.9. There's a lot of correlation between the traits. In the end, we kind of have to do some kind of selection. Which traits are important to look at structural diversity? One way to do it is to apply a principal component analysis and look which are kind of the major axes that are important. So which traits do we choose? In this case, I decided for one height-related metric. So we see that all the height metrics are kind of clustered in the principal component space. So I choose relative height 50%, also because the maximum canopy height is more strongly correlated with foliage height diversity, so I want to have a bit more independent traits. Then there is two density-related traits which are very important. That's the total plant area index and the plant area index from 0 to 10 meters, so kind of like how dense is your understory. And then for the layering, there is also two traits that kind of stick out as individual components in the space. That's canopy ratio and foliage height diversity. So I didn't pick the traits which were best measured by GEDI in this case, but the ones which I thought were like most relevant and most independent of each other. So you can get probably better results if you would just pick the maximum height and 2-3 others, but I was more interested to see in a realistic case of traits that we find are actually ecologically relevant. So how do we go from the traits to diversity? In this case, I use, basically I'm looking at the distribution of a certain area of the traits in the trait space. So you look at the probability density distribution. It's like histograms in the 1D case. And then look at functional richness is the coverage in the trait space. So if you have a very rich ecosystem, this histogram or distribution might be very broad. And you can extend this concept from 1D to 2D and n-dimensional basically. So in our case, we have 5 traits. So it's a 5-dimensional space, and we basically look at how much does this ecosystem cover in this 5-dimensional trait space. That's functional richness. Then I'm looking at functional beta diversity. So that's kind of like how different is one area to the other. So you can overlap those trait spaces, and functional beta diversity is basically 1 minus the overlap. So how much unique trait space or unique characteristic does an area have compared to its neighboring areas? And just to illustrate that, if you have like 2 pixels, here we have very high functional richness, but not much turnover between the sides, so beta diversity is very low. Here we would have like low functional richness, but very high beta diversity. There's basically no overlap between those niches or trait spaces. And the final example is kind of like high functional richness in 2 areas, but also a high beta diversity. That's something we would expect, for example, along environmental gradients where there is high alpha diversity and high beta diversity. So now to the results. How does it look like, those patterns? So we take the airborne result as our reference because it's wall-to-wall coverage. It captures the full diversity of the forest. So we see that we have high functional richness along these main valleys. Then we have high richness where we have like the more diverse forests here with also the very tall sequoia trees and more diversity of different structure types. And then it's not surprising that towards the really high elevations on the eastern side, we have less vegetation cover, but also less structural diversity. So here in the middle, we see the result, the same mapped using the GEDI data simulated without any cloud cover. So it resembles the patterns really well. It's just a little bit more noisy, but in general, we are really able to map functional richness in this area. And if you look at a simulation including cloud cover. So we use the cloud frequency data set, which basically tells you the probability of clouds for each month. So we can just reduce the number of GEDI shots based on that. And we see that we have persistent cloud cover, but again in the very high elevations where there's actually not much vegetation. So we get those gaps here where there is hardly any vegetation or functional richness, and some kind of scattered gaps, but they don't influence the results much. It's a little bit different looking at beta diversity. Beta diversity is a little bit more difficult to predict. We see that we have, again, high beta diversity at those transitions between canyon and higher elevations, but also in those parts, higher elevation, the vegetation gets a bit more patchy. It's also very dry. So you have pretty high beta diversity because the structure changes a lot with like local availability of water or moisture, probably also depending on snowmelt and other factors. This is again captured pretty well by GEDI if we don't have cloud cover, but if we lose like number of samples, we get in many parts an overestimation of beta diversity. That's because we cannot sample the full trait space, so there might be actually some overlap, but because we only have very few samples, it looks like the areas are very different and actually don't overlap. So we have like some overestimation of functional beta diversity. So if we just look at the difference maps, this is the difference between GEDI and airborne functional richness and functional beta diversity. We see a slight underestimation in areas where we have taller trees and more tree cover. That's again expected because we are subsampling the landscape. We don't capture the full diversity. And we have a little bit of an overestimation in those higher elevations. That's because we have very low vegetation and very steep slopes. So if the ground energy is kind of elongated, it sometimes might look like a tree or diversity, but it's actually not. So that leads to this kind of overestimation here. And now if we switch to the case with cloud cover, it's looking pretty similar for functional richness, so that's not that much affected by the reduction in number of GEDI shots, whereas beta diversity is. And since there is pretty clear patterns of underestimation and overestimation, I thought maybe it's possible to explain those differences by just looking at slope, topographic diversity, and tree cover. So I built a very simple model, and it predicts like about 55% of the variance. So we can use that to kind of build a hypothesis where we would expect GEDI to potentially overestimate or underestimate diversity. So this is all of California and our study site. So we can just basically have this simple prediction. It's not a quantitative way, it's more qualitative and more hypothesis of where we might expect some underestimation, where we have a lot of tree cover and some overestimation of diversity in areas with low vegetation but very rugged terrain. And we can refine that a bit by actually excluding the areas which are not vegetated at all, so these are just deserts, and adding again the cloud cover, and we see that we would do pretty well in many regions, but have like some slight underestimation where there is more tall trees and some potential overestimated areas in, for example, grasslands with very rugged, steep terrains. To conclude, most of the traits derived by GEDI have a pretty good R-square value. Especially the height-related variables. It's not surprising that relative height 25 percentile, so kind of understory related, is more tricky because of the topography of this area. And functional richness
works well to map. It's also not too much affected by the density of GEDI shots, whereas functional vector diversity is. So we would have to probably define some kind of threshold, what we think is the necessary amount of GEDI shots we need to be actually able to say something about functional vector diversity. And I should mention that this was all done at the 1 square kilometer scale. So with that, I would like to thank the GEDI science team, the NASA Airborne Snow Observatory who provided the airborne data, and the JPL Carbon Cycle Group and the University of Zurich Research Program on Global Change and Biodiversity. Thank you very much.